Cancer is characterized by out of control cell growth, and it becomes especially harmful when these altered cells form lumps called tumors. Less dangerous tumors are called benign tumors. However, more dangerous ones, called malignant tumors, can spread cancer cells to other parts of the body. The best way to prevent this from happening is by removing the tumor before it can spread. It's important to completely remove the cancerous tumor to prevent cancer recurrence, resulting to decreased survival. Current methods of diagnosing and checking cancerous cells, called frozen section, require extracting a small piece of tissue, and it takes 30 minutes, prolonging the operation and may have inaccurate results. Molecular analysis of cancer tissues offers an opportunity to incorporate cancer specific biomarkers in clinical decision making for improved cancer detection and diagnosis. Mass spectrometry imaging approaches have been successfully applied for molecular imaging of cancer tissues, including rapid evaporative ionization mass spec using a tool called the iKnife. A group of researchers at the University of Texas at Austin, led by Livia Schiavinato Eberlin, designed and developed a device that can accurately detect cancer in seconds during surgery in a non-destructive manner, which they call the mass spec pen. And unlike previous techniques, which require tissue damage to produce molecular ions, the mass spec pen allows real-time, non-destructive sampling and molecular diagnosis of tissue using mass spec techniques on a discrete water droplet. The mass spec pen was designed to minimize tissue damage, maximize tissue analyte extraction, and maximize solvent transfer. The main component of the handheld device is an inexpensive and disposable 3D printed PDMS tip. It was designed to have three ports: an incoming port that delivers a single water droplet to the probe tip, a central port for gas delivery, and an outgoing port to transport the droplet to the mass spectrometer, all connected to a reservoir with a diameter range of 1.5 to 5 millimeters. A surgeon would hold the pen against the tissue. The incoming port delivers a water droplet of fixed volume to the tip or reservoir, where water soluble molecules on the tissue, including cancer specific metabolites, would dissolve into the water. After a controlled of time, with an optimal exposure time of 3 seconds, the water droplet is pumped to the mass spec through vacuum extraction. The gas delivery port delivers low pressure gas to prevent the collapse of the system due to vacuum, and to assist solvent transport from the tissue to the mass spec. The mass spec was cleaned through a rapid, automated flushing or by replacing the disposable tip to avoid contamination. The droplet containing the molecules is pumped to the mass spectrometer, an instrument that can identify molecules based on their masses and the trajectories they take through an electric or magnetic field. The mass spec can then provide information on each molecule present in the sample, such as its mass to charge ratios, which is unique for each molecule. Their results show that there is, in fact, a difference between the molecular composition between normal tissue and cancerous tissues. The information is analyzed by a statistical classifier called LASSO. They train the algorithm on different tissue samples to recognize cancer biomarkers based on their mass to charge ratios. Using this, the tool can display whether a tissue is normal or cancerous, helping the surgeon decide which tissue to remove. It can also predict a cancerous tissue's histologic type. They tested the pen on 20 human cancer thin tissue sections and 253 human patient tissue samples, including normal and cancerous tissues. They reported results with an overall accuracy of 96.3%, high sensitivity of 96.4%, and high specificity of 96.2%. The mass spec pen can be compared with a similar device called the iKnife, developed back in 2013, which at the time was a very breakthrough invention. The eye knife is similar to the mass spec pen in that it identifies whether or not a tissue is cancerous within seconds. It also uses a mass spec to identify cancer biomarkers. However, it differs in the way it extracts the samples. The eye knife is an electrosurgical knife that vaporizes the tissue, creating a smoke that is sucked into the mass spec to be analyzed. Because of this, tissue damage is actually required before you can get the diagnosis. The mass spec pen is able to do the same with similar results and accuracy as the eye knife without destroying the tissue. The research group is working on a software that would spatially map the results to assist the surgeons further, so that they can opt to remove the tissue in one go rather than in pieces. With their current technology, they can also create other databases for diagnosis of other diseases that are difficult to test for, following the similar, if not the same, extraction process. My idea is to integrate their technology in a multifunctional tool, if possible, one for laparoscopic surgery, a tool that can have several attachments, one to probe for the tumor, another to test whether it is cancerous or not, similar to the mass spec pen, and another for extraction or removal of the tumor.